Hello, everyone. Thank you for joining us for today's seminar. This is the Statistical and AI Methods for Health Data Science seminar series organized by the Startup AI Alliance. So here just a brief introduction of the group. So the group aims to organize activities that empower institutions to participate more actively in AI research and leadership, including this webinar series. And then here are some additional informations. And then also right now, the group has been officially recognized as an ASA interest group. Here is the website, and then we also post seminar recordings on this YouTube channel. Here's the committee members and then the other officers. And then today it is our pleasure to have Dr. Dana Tudorascu, who's an associate professor of psychiatry and biostatistics at the University of Pittsburgh, to talk about harmonization in pattern and MRI imaging. And then Dana is also a co-leader of the study design and data analysis group and the leader of the Biostatistics group for the Alzheimer's disease, for several Alzheimer's disease programs, including the ADRC at Pitt. So she made a lot of contributions in terms of data harmonization for neuroimaging data, brain tissue classification with wet matter lesions, and then AD research. And then before handed to Dana, just some logistic of the webinar. So if you have any question, please, during this presentation, please type it in chat. I will monitor it. And then if it is closely relevant to what is presented, so then I will just maybe ask Dana to pause and then respond to the questions. And then if we have time, so you can also save the question to the end of the presentation, and then we will have some time to discuss and then ask the questions for Dana as well. And then, hold on a second. Let me stop sharing and then hand it over to Dana.
Okay, well, thank you so much for the invitation. I am actually very excited to show you some of the work that we have been doing. I think what you guys are putting together, it's very, very good. And I was just saying a little bit earlier how I wish the students, a lot of students to be here. So let me make sure that I can see this very well, minimize some of these. Okay. So I will talk about statistical challenges in data harmonization studies. In recent weeks, we started to work a little bit on blood biomarkers of Alzheimer's disease, and I will show you a little bit about that, just a tiny introduction. So an overview of my presentation is as follows. So I'm going to talk about the biomarkers harmonization. I'm going to give you some background methods, and I'm going to present some of blood biomarkers, which was not in the title, but I thought we've done some preliminary analysis, and I really wanted to share with you all to see what we found. And then I'm going to talk mostly about PET imaging data and harmonization methods. Some of the results from the head-to-head studies, some of our new phantom studies, which we have started, and I'll give you a summary. So the motivation behind this work, and thank you, Yi, for such a great introduction. I've been co-leading a lot of these multi-site studies, and I think a lot of my work is based on the fact that, before we try to analyze the data from just one study, we didn't have as many challenges as we have today. But some of these data is acquiring, all these images on different scanners, and not only images, just different data domains, cognitive data, blood biomarkers data, proteomics, metabolomics, all of it from different sites, different scanners, different machines, different assays. So it's very challenging to analyze them. So one of the studies that I'm going to talk about today is this head-to-head harmonization of tau tracers in Alzheimer's disease. And University of Pittsburgh, including myself, were also part of the ADNI, and especially in developing a single universal scale for tau tracers. And I will talk a little bit about that. And then the other thing is that, you see all the names of the studies, but then one of the major challenges, I said, is that working with this data from all over multi-site studies. So what I wanted to show here, is a picture is worth 1,000 words. Is that when we look at the data, we don't look at very nice data, especially from these multi-site studies. And What we thought it was solved before, we just realized that maybe it wasn't, and maybe we didn't understand it, or maybe the methods that we thought were going to work aren't actually working. And one such example is, if you look at this, you can see in this right panel right here, which is called amyloid centiloids on the same subjects of different PET scanners. So this is actually a method which was called centiloids, and it is for amyloid imaging, and is to standardize tracers across to standardize amyloid imaging tracers. So we thought that this problem was solved. So transforming the amyloids to the centiloids, the distributions would actually overlap. However, these were studies that started in about 2009, but we have never done this in the same subject. So we scanned the subjects with different amyloid tracers, and we use this centiloid scale, which I will be talking about. And we thought that the data is harmonized or standardized. However, if you look here, you can see that it's not actually the case. So this is something that we have just seen in recent months. And I wanted to make you aware of it for all of you working with amyloid, SUVR, and centiloids data that might not be necessarily harmonized. The other thing that we started looking at is blood data. So blood data is p-tau217 is a biomarker for Alzheimer's disease, and it's starting being acquired in several studies across multi-sites with different assays. So in the tube, you get a different assay, and you collect this blood, and it's a one outcome per subject. But what happens is that if you use a different assay, and these ones here, there are 4 of them in the same subject, the distribution is quite different for all the data. And then the other thing is, when you collect data on MRIs, the same subjects on different scanners, the distribution should look similar, but it doesn't. So that's why we kind of need these methods of harmonization. So this is sort of like the motivation behind all the work that we've been doing. So one of the major challenges in Alzheimer's disease that I've been working on for a while, and every time I give a talk on PET, I feel like it's an infomercial about trying to get more statisticians involved with PET imaging, because there is so much data, and there are so many problems that we can contribute to actually make these multi-site studies more standardized. So we have advanced right from one-site studies to these multi-site studies, and we have images collected on different scanners, different manufacturer, different strength, PET, MRIs, and then we have longitudinal studies where you collect data on different scanners at different time points, at the same site or at multiple sites. And then in PET imaging, there is another problem, which is tracers. So, and I'm going to give you a lot of background on that. So we have images that are collecting using multiple tracers. The challenges are not only that, but we analyze data with different software, different pipelines, different platforms. We use, the derivation of the imaging outcomes could come from multiple sites. So I can tell you from the ABCDS study that we have analyzed all the data from the ABCDS at the University of Pittsburgh and at Mayo Clinic, and we have found about 40% differences in cortical thickness, exactly the same pipelines, the same subjects, but different labs. And then, as I said, the different sites with the different tracer, which is a major problem at this specific time point, at this specific point in time, especially in Alzheimer's disease studies, because of the clinical trials that are using these different tracers. Right now, the current standard in PET imaging, data harmonization, it is actually the smoothing, and it doesn't necessarily work. And as I just said, centiloid standardization was previously thought to standardize amyloid, but we have found that actually it doesn't. I'm not going to show you the data. It was just that plot, but we're working on that. And then in recent weeks, we were asked to look at some correlation between blood biomarkers and imaging data. So we said, okay, so let's just look at this blood data to see how it looks like. And we found that it was quite variable. So one of the tau proteins that are used as a promising tool for diagnosis and disease progression in Alzheimer's disease is the phosphorylated tau, which is p-tau 217. Now, I am an imaging researcher, right? So this is just very preliminary for me, and I am just learning at this point about these blood biomarkers. But there is one thing that we know, that these are very easy to collect. They are very easy on the patient, and they are also very low cost. But the challenging are that, the blood is collected using different assays. It is unknown whether these levels can vary during the time of the day where the blood was collected, and processing variability could be due to the method of the extraction, the calibration to a control group, because that's what they usually do. They calibrate each blood sample to the control group. Now, if you look at this, so we just said we have just done very simple preliminary analysis of these blood biomarkers, and I wanted to show it to you for those of you that work with it, or for those of you that are interested in working with it. I think there is potential here for methods development, and this is just one of them, the p-tau 217. So you can see that if you look at these means, these are. So one thing that I wanted to tell you is that this data is using different assays, but these are the same subjects, okay? So these are the same subjects measured with 4 different assays. One is LZ-PAT, C2N, Jensen, and Lumipulse. And if you look at the means across these assays, that you can see that there is a huge difference between these means. And also you can see the distribution of the variability. This is a sample from ADNI. There are 208 controls, just 10 dementia patients, 174 MCIs, and 2 that were unable to determine. So a student that is working with me, so she has done some investigation and just looked in the literature to see what methods are out there and what methods people are using for blood biomarker harmonization methods, and we just applied them to the data that we had, okay? So one of the methods is just called simple means. It's a mean normalization, it's no variance adjustment. Another one is standardized batch mean using linear regression. So you basically fit a linear regression as a function of batch and covariates. Quantile normalization and quantile regression. These are fairly, these are no methods in statistics. There is nothing fancy here about what we are using. However, we said, okay, so let's just look at some of this to see if the data looks the same. So what you would expect the distributions, you would expect the mean values, the median values to be closer, or the distribution to be closer. But we had quite a surprise to see that that is actually not the case. And you think, when you think about imaging, that's much more complicated, right? You have so many variables, you have high dimensional data, but this is also a challenge because you only have one variable. So the standardized, I'm just going to show you results from just 2 of these methods. But if you look at the standardized batch mean using linear regression, it didn't actually do much. There is some closeness in the mean, but the variability still remains the same. The medians are also different. So that didn't actually work. One of the things that, you would expect that to work, because usually quantile regression and quantile normalization are known to be able to fix this kind of variability across distributions. So this is actually did work better. As you can see, the mean of these 2 were generally closer together, and the mean of these 2, so the LZ-PAT and C2N are closer in Jensen, and the other one, they are also closer in value. So this one does, a pretty good job in closing the, the variability gap between, across these methods in the mean and in the median. So, but then when you look, so one thing, I think one caveat, and as I said, we just started working on this. So when you look at these measures and when you look at these methods, we haven't done it for each group separately, for each disease group. So we've done it overall for all all the disease groups together. But then we separated the results based on that. And there is more work to do here. However, when you look at the differences, between the raw data, which there was no massaging done to it, or no standardization or harmonization, you can see differences right across. Let's just look at the MCI, for example. The dementia patients, there were too few. And then the controls, also huge differences in the raw data. The simple means, as I said, it's getting the means together, right, closer together, but the variability doesn't do anything. The standardized regression also, just seems like the LZ-PAT and the Jensen are closer together, and the C2N and the Lumipulse are closer together. Other methods are showing different things. The quantile regression, that's the one that is normalizing, and the quantile normalizations are two of the best ones. But as you can see, the LZ-PAT, C2N closer together, and these two are closer together. But when you think about it, one thing that we haven't done yet, but one thing that that we always do is to find out what is the sample size for these studies, what are the effect sizes. And you can see that depending on the actual assay method that you're using, even if you do a harmonization or standardization or whatever you want to call it, you still have these differences. So when you calculate effect sizes based on these numbers, you are still going to have difference in the actual sample size estimates. Of course, if the variability, if the differences in variability is not that big, and then, you don't get such huge differences, but it's very method dependent and assay dependent. So when you think about these multi-site studies, if you're collecting data with different assays at different sites and you want to put it together, then that becomes very problematic. So as I said, I just wanted to show this as one thing to, something to think about us as statisticians when we work with this kind of data, when we work with an investigator, when they give us some of these summary measures, we usually have to ask, where is this coming from? What type of assay has been used, and all of that. So I think that there is a lot of background that we actually need to learn. So I think one of the next steps in this blood biomarkers is that we have to analyze, probably do all this harmonization within group. I have seen a recent paper that showed that the Z-scores is actually good, and we know that the Z-scores are doing a good job. I haven't investigated that so far. I think the effect sizes and sample sizes need to be computed for different methods, and I think we also have to look at further associations. And besides the p-tau 217, there are several other biomarkers, and I think a lot of the NAC has some of these data that you can look at. So I have mostly looked at the one in ADNI. So I want to go now get into the imaging stuff, mostly in the PET imaging. And I'm going to start with a little bit of the background. Some of you may have seen some of this stuff that I have presented before, but I think it's really, really important to understand what is happening in the background of the PET imaging, because it's one of the imaging techniques that is much more complex than the others that, such as fMRI and MRI, especially in my opinion. So I've been working with this for close to 15 years. And there is still a lot that I am learning every day from my collaborators. So what you see on this side is, on this slide is, when you image using PET, you have to inject a radio tracer compound into someone's vein, using some sort of an arterial blood or some sort of an injection, right? So you put the person in the scanner, and then, you do this injection, and then you have to image within a certain period of time. So what happens here, right here in this plot, what you can see is that you inject the radio tracer, and then the radio tracer has to cross the blood-brain barrier, and then it starts being accumulated. So what you see is a region which is called posterior cingulate, is one of the areas that in Alzheimer's disease we use as as one of the areas that accumulate some of these tau proteins or amyloid, and we see we look at that measure specifically. Now, we use the accumulation in the region, okay? So this is how it accumulates over time, and then you calibrate that to the cerebellum. Cerebellum is known in these studies to not accumulate amyloid or to not accumulate tau. Now, FTG is a different, it's a different thing. But for Alzheimer's disease, the cerebellum is used as a calibration and is used to divide by, so the ratio that we extract is called standardized uptake value ratio. So within this specific time frame, you calculate it, they use kinetic models, and we usually get the data processed in terms as either voxel images with SUVR or summarized images. So right here on this side of the slide, you see the MR image, and then you see the PET image. And this is 11C, it's Pittsburgh compound B. This is amyloid that is used to measure amyloid in the brain, but you need an MRI in order to co-register because the PET resolution is not good. Then on this other side of the slide, you can see, there is amyloid, and then there is tau, fluortaucipir, and then there is MK 6240, and those are measuring the tau protein in the brain. So there are differences in these two tracers, and I'm going to talk about that. But what I wanted to say is that this is kind of like how the images look like, and as I said, you need an MRI to be able to see the areas of deposition. So what I'm going to talk about are two tau tracers. One is MK6240 and the other one is FTP or AV1451. They are used to measure tau proteins in the brain or tau accumulations. So tau areas are categorized into BRCA regions in the brain of people with Alzheimer's disease. BRCA 1 and 3 are considered to be early accumulation, and 4, 5, and 6, there are later regions, they are more advanced in the disease. And as I was saying, the standardization or the tau PET measures, the SUV is called the standardized uptake value, and is measured by averaging activity concentration in the brain, is divided by the, the actual individual injected dose, so it's calibrated by the body weight. And then the SUVRs are the standardized measures that we use in PET analysis. We sometimes use SUVs as well, and that is calibrated to the cerebellum. Okay, so one challenge of the PET harmonization is not only the scanner, but is the tracers. The goal of the tracer harmonization is that tracer values and distributions should be similar, because they are using these in clinical trials. The challenges are that in tau imaging, you also have this in amyloid, but not as much. But in tau imaging, you have something that is called off-target binding. So what happens is that the tau accumulates in areas of the brain that have nothing to do with the disease, but they are also individually varying by sex, by other individual differences, and this could artifactually increase or decrease the SUVR that you are using as a measure to do your statistical analysis. One of the harmonization methods that have been emerged in PET imaging for tau tracer is Centaur-Z, and it was developed in 2022. So on this slide, you can see, so this is kind of like the same subject that was measured with two different tracers. And what I wanted to emphasize is that you don't need to know PET imaging. You just can see that there is a difference between the FTP tracer and the MK tracer. And these are areas of off-target binding. Choroid plexus area, the striatum, as you can see, is lighting up here, but not here. And then the meninges, which is this round, the wrinkle around, the ring around the brain, which is more prominent in MK6240. However, what we have found in recently is that it also varied by scanner, because there is a lot that goes back into the tau processing. So we found in some sites we do have meninges that is lighting up in MK, in some sites we do not have that. So that becomes another issue. So what I was saying before, I said that head study is the one that I was going to show some results for. So why head study is so important? Well, it is because it's one of the first studies where you have subjects that are imaged with more than one tracer. So in this presentation, I have 2 different tracers, but we are up to 4 different tracers within the same subject. So each individual was scanned. Here is just 3 scanners. We have 8 or 9 sites in the head study. I think 1 was recently added in Europe. And then each of these subjects were scanned a few months apart with a different tracer. Now, the thing is that the amount of radiation that a person can take, it has to be with a certain limits that were actually approved by the FDA. So if in MRI you can have 10 images, you can have 10 scans of the same person and you can do a traveling study. In PET imaging, you cannot do that because you are very limited by the amount of radiation that you inject into a person's blood. So here there are 2, so each person got 2 different tracers a few months apart, and I believe that in year 2, we're adding more tracers, so that was enough for 1 year or several months. So the sample composition is, cognitively unimpaired, mild cognitively impaired, very few AD subjects, males and female distribution quite similar, and in terms of age, we're going from the minimum of 21 to a maximum of 87, and with a mean of 62. What I want to draw your attention here towards is the fact that when you look at these plots, what you want to see is you want to see them on top of each other, right? You want to see them overlapping completely, but they are not. As I said, we've had studies before where you would scan different subjects at different sites with different tracers, but this one is the same exact tracer, measures the same exact thing in the brain. So they are quite different. So this is for just one BRAC area. So you can see the distribution of the FTP that is in red, and the distribution of the MK. This is for all the subjects. If you look at the B1 and the B2, so this is MK in BRAC5 by the diagnosis group. ADs are in red, MCIs are in green. Unimpaired are cognitively unimpaired are in blue. And you can see the differences between these plots between the MK and the FTP. And again, I'm emphasizing here mostly tracer differences, not scanner differences. And then, across the sites, these are different sites. You would expect that because of the different composition. As you can see, again, 3 different colors for 3 different sites. Now, if you just look at this one, which is highlighted in here, these are the normal controls for all sites. The FTP, the SUVR is around 0.50, and the MK is 0.35. Now, if you look at across the sites, you can see that the FTP is actually higher compared to the MK. And these are the actual normal controls where the sample size was a little bigger than the other ones. But this is not always the case. However, there are huge differences between these numbers. They're almost close to being, 1 twice the value of each other, and that is very problematic when you use different tracers across different sites. And again, if you see this in the same subjects, think about what you would see when you use different, radioactive tracers. Now, in PET imaging, same as in fMRI, same as in MRI, same as in other imaging technique, you have voxel level harmonization, and then you have summary measures harmonization. So I think the statistics field has done pretty well with the summary level harmonization. The Combat, JP's, and Taki's group paper is very, very good at harmonizing imaging measures, summary imaging measures, not so much for the PET, but has been doing very, very well in MRIs and other imaging areas. So what you're looking for is these summaries, these image summaries, to be close in values and in the distribution to overlap. But we've seen here with the tracers that's not necessarily the case. And in voxel level harmonization, you're not only looking to make the images more alike, but you also want to preserve the anatomical structure. Is those boundaries where we see a lot of differences. So you want to preserve that, preserve the functional activity. And right now, I think the state of the art PET harmonization technique is smoothing, and more recently, as I said, the Centaur Z method, which I'm going to talk about. So this Centaur Z method was developed around the idea of the centiloids. So centiloids harmonization was developed for amyloid. Centaur Z is for tau tracers. Amyloid images, amyloid plaque in the brain. Tau measures tau proteins in the brain, but it's kind of like the same idea. They measure the same type of activity, standardized uptake values, or amyloid or tau deposition. So the centiloid harmonization was a quantitative metric that was developed at the University of Pittsburgh, and it was developed in a sample of individuals, 34 young controls, and about 45 individual AD patients. So they took the maps of these patients, they overlapped them, and then they calculated the centiloid scale for each individual. You subtract the average of the controls, and then you divide by the difference between controls and AD, and then you multiply by 100. Now, the Centaur Z was supposed to be developed along the same lines. However, what they took was, because you wanted to harmonize across the tracers, so they took these different patients that were scanned with different tracers. They created an overlap type of map, so something like a universal tau mask, and then that was co-registered with the MRI and the gray matter, that you have to derive the regions from that. And then 4 subregions were created with certain constraints within this universal mask. And then for each tracer, such as mean and standard deviation, some Z-score values were calculated. Okay? So what happens is that, so you derive these conversion equations from the SPM or whatever measure you're using, and then you can calculate these standardized Centaur Z values. So depending on the tracer that you're using, okay, so for each tracer, there is this universal mask. So for each tracer that you're using, you substitute this X value, and then you get your measurement for the specific tracer, specific tau tracers, to convert it to this universal centiloid scale. The paper has been published, but what I want you to see is that you can see that all of these assume that there is a linear relationship between these tracers, okay? So again, this was only shown across different tracers and different people. It's not the same people with different tracers, but different sample of subjects, and then these conversion equations were derived. So we have done that. We have applied the Centaur Z harmonization to our data, where we had head-to-head subjects. So we had the same subjects that were scanned with 2 different tracers. So for example, I want to draw your attention to this quadrant right here. This is a 76-year-old female subject. This is an AD, and using AV or FTP and using MK tracer, you can see that the Centaur Z value, the universal Centaur Z value, was 16.63 with the AV, and it was 25.54 with the FTP. So they are not harmonizing the data within the same subject as all. The values are very different. The same with the SUVRs, right? 2.38 and 3.53 right here. There is also a huge difference in this one. It's almost double, right, in the other side. But one thing that I was saying before is this off-target binding. So if we look at site 1, if we look at the control subject, it's a 58-year-old woman control, you can see that at this site, the MK, it has the meninges that is lighting up. So it's probably interfering with this SUVR value of 3.53. However, at this site, that is not happening. So in this scanner, you don't have the interference of the MK. You don't have that off-target binding, so probably this value is more accurate. But there is still a huge difference in the controls, and there is a huge difference in the AD. So Centaur Z harmonization doesn't actually work, and this is the best that we can get, where you have 2 different tracers in the same people. And it's not supposed to be due to the disease, because the amount of time that have elapsed between when you scan it the first time and when you scan it the second time with a different tracer wasn't enough time to create this accumulation such that the numbers are so different. So it's all artifactual increase. So it's not there. It's something that is causing this, these values to be so different. Now, as I was pointing out before, so the equations that have been developed, they are all linear equations. But when we look at this data in the same subject, you know, with the FTP and the MK, they demonstrate a potential nonlinear relationship and diagnosis dependent, because so this on the y-axis, you have the MK, the center Z values, and then the x-axis, you have the FTP. So when you look this, you can see that, so right here in this quadrant, you probably have all the controls because they have lower values, but then you go up on the subjects that you're most interested in, where there are your MCIs probably and your ADs, you can see that the relationship doesn't seem to be linear. Is it true that the value, the samples are not that big? However, we have seen this when we increase the sample values, which I do not have here. So it's very questionable whether the center Z is actually an acceptable method of tracer harmonization, right? But we also look at the effect sizes. So before and after combat harmonization at the level of the BRAC regions, what I was saying. So now, one thing is that, so BRAC area number 2 is a very controversial area in Alzheimer's disease. And why is that? In BRAC area 2, there is a lot of off-target binding. So when you look at these effect sizes, okay, so the effect sizes are the ones that are not in parentheses. And then the effect sizes are between the CU and MCI, CUs and ADs, and AD and MCIs. So what you are seeing is that these sample sizes are very variable, and they are based on the tracer. So if you use, for example, CU versus MCI, BRAC area 5 between MK and FTP. So if you use the MK tracer, you need about 20 subjects for 80% power for detecting these differences between the two groups. But if you use FTP, you only need 34 subjects. Now, well, maybe it's not such a big deal, but when there is $5,000 or $8,000 per PET scan, that makes a huge difference, and especially in the clinical trials, right? This is for the harmonized data. Now, if the data is not harmonized, now, as you can see, there is quite variability between the effect sizes. Now, the non-harmonized data give you totally different numbers. If you look at the BRAC5 in MK, that you need 52 subjects. With the FTP, you need 34 subjects. And again, this area, BRAC5, it's one of the more advanced areas of Alzheimer's disease. So if you were to monitor, maybe you want to go at the areas that are, you know, either at the area at the end that is a BRAC5 to see if there is a treatment effect, maybe at the area of the beginning. And in BRAC2, where there is a lot of off-target binding, you can see, you know, again, a lot of variability. So the data that we are getting from this head-to-head study shows a lot of variability in terms of the statistical analysis, in terms of effect sizes, in terms of the relationship between the tracers, in terms of everything that was done before. Now, when we are looking at these within subjects, between tracers, we see how problematic everything is that we have said before, that it was working, that we thought we had it down, that we thought that the methods work. It's not necessarily that, okay? So what are we gonna do? Because the situation is that we don't know what the actual truth is, and it's very hard to tell what the actual truth is. So we're gonna go back to the phantom studies, okay? So some of you may say, well, but Annie has phantom studies. Yes, and I'm going to show you what is wrong with them. So how are these multi-site studies actually calibrated, right? We don't know the ground truth. So Annie's study has what is called the Hoffman phantom, which is a traveling phantom. Now, Hoffman phantom has a single chamber that is representing the whole brain. And as I was telling you before, tau and amyloid, they need cerebellum to calibrate the scanner. But Hoffman phantom doesn't have a cerebellum. It's just one single compartment, brain, right? And that's why it doesn't actually work. And there were recent studies that have shown that Hoffman phantom works for the FTG. Now, FTG is fluorodeoxyglucose. There is a different type of tau tracer. It's used a lot in cancer. It binds to sugar molecule. It doesn't need to be calibrated to the cerebellum. So it's a whole different story whole different behavior of a tracer, but we're using that for scanner calibration in ADNI studies, right? So what we did was, okay, well, University of Pittsburgh is part of ADNI, and we had Hoffman phantom data. So we said we wanted to look at it. So as I said before, smoothing is the standard for these studies and to capture scanner variability. So we have two scanners at the University of Pittsburgh, HR Plus and MCT, and this is the Hoffman phantom template. So we looked at the data that we have collected, and you can see on the HR Plus, the SUV was 4.72, and this is 4.54. Now, if you smooth it, as I said, these values should become the same, right? So, and now it's 4.37, 4.10. So it doesn't do much. It doesn't bring them together. So the actual difference, it remains the same. However, you cannot compute SUVRs because the phantom is a single brain compartment and doesn't allow the cerebellum. Well, but you must say, right, but the cerebellum, there is nothing that is deposited in there, right? So even if you calibrate it to a cerebellum, the difference couldn't be that hard, that much, right? So what we did was we said, okay, well, let's just look at the phantom that is calibrated to the cerebellum, right? So my colleagues here at the University of Pittsburgh have developed a phantom that has multiple brain regions, and it also has a separated cerebellum. So what you see here is sort of like mimicking the areas of the brain. So all these little channels, they are one area of the brain. The thing is that they are separated, so you can control the amount of substance that you can put in this phantom. So we were able to actually, not myself, but my colleagues, they were able to control. So you're able to control, and you know exactly how much amount of this radio tracer you put in these chambers, and you scan it at the same time with the cerebellum. We scan it on two different scanners, and we were able to control the amount of substance. And so that you can create a control subject, and you can create an AD subject. And then, because you have the cerebellum, you can calibrate and you can get the actual values. So one other thing is that they also, before they put all of this, all of the substance into these areas, they also scan the actual bag with the concentration. So they knew exactly, it was a very controlled study. We knew exactly what we were putting in the phantom, and we knew exactly what we got out. So, you wouldn't expect to see any difference. This is a phantom, right? Two different scanners, everything very, very controlled. However, when you look this, this is some sort of like a mesh model, and then this is the CT scan, and kind of like the MCT scanner and the HR Plus. As I said, it's just mimicking different regions. So we had 4 different regions here, temporal, medial, and superior. But when you look at these values, so these ones right here, okay, so this says MCT and HR Plus, this is 8 millimeter, this is smoothed, right? And this is the cerebellum. Okay, 11.08 and 9.4 between the two scanners, very, very controlled. The cerebellum right gray matter, 10.7, 9.8. The left gray matter, 10.7, 9.9. Now, if you look at the smoothed images, again, the standard method of harmonization, you can see 0.96, 1.04, 1.03, 0.997, 1.31, 0.95. Again, these aren't big differences. However, this is a phantom, and there shouldn't be any difference. It's not even this much, right? Then if you look at these areas that are more important, you can see this is 0.889, 0.871, 0.65, 0.64, 0.442, 0.429, and these are areas that we are interested in Alzheimer's disease, right? So in terms of differences with, we used harmonization, and specifically with and without the smoothing, what we have seen in SUVs, right, not calibrated to the cerebellum, and then SUVR differences. So if the smoothing works, we should see no difference between the two scanners. However, we have seen not such big difference between the no smoothing versus the smoothing in the SUVs or in the SUVRs. So specifically in the SUVR, you can see that the differences are actually somehow, this is actually bigger in the mid-temporal than it is before smoothing. And in the inferior temporal, this is bigger. And then it's a little smaller after the smoothing, but it still varies between, 1.4 and 2 to 3%. So I think the amount of times that we scanned the phantom was 8 times in each scanner to be able to get this variability. But the idea is that the cerebellum, which is supposed to be a very stable region, we have seen differences between the two scanners, and that is something that is very worrisome, and especially, and this was just one tracer. So the phantom studies only capture scanner differences, right? We cannot capture differences in the tracer. We cannot capture individual differences that are due to biology of the patients. So if in such controlled environment, we can see these huge differences, what do you guys think are we seeing in these multi-site studies? So as statisticians, again, I cannot stress this enough, is that when you work with data, you have to really pay a lot of attention. Where is it coming from? How was it processed? What was it used? So I think one of the next steps would be to extend some of these, the longitudinal combat to account for these different tracers within person, but then you have the off-target binding that has to be taken into account into that model. We do have some ideas on how to do it, but I think it would be a very complicated statistical model. The other thing that we are in the process of doing is basically analyze data separately for each scanner and each site in the human studies, and then combine them using meta-analysis. We have done harmonization studies in MRIs, and the papers are out there. You can go and look for them. One of them is superpixel combat harmonization to accommodate different scanners within the same person. So that would be another thing to try, but we haven't tried it yet, and we don't know if that's going to work for the PET, because it's a totally different beast than the MRI is. And one of the most important things is that we have to take this brain phantom on multiple sites. This is part of a grant that we put in, and it hasn't been funded just yet. Hopefully in the future. But I think the actual brain phantom is something that mimics as close to the truth as we can in terms of scanner variability, but the individual variability is something that you have to add. And how would you add that? How do you account for these individual biological variability to capture the actual differences? So in summaries is that, we show large differences between scanners using this phantom, and we shouldn't be seeing that. And this is the first time that something like this was done. And you would think that we put billions of dollars into all these multi-site studies, but we don't have something that is actually collecting the truth, right? What is the actual truth? This tracer harmonization is very challenging. I know that there are multi-site studies that are collecting different amyloid tracers at different sites using centiloids harmonization. So I told you that doesn't quite work as well as we previously thought. So that's a problem. And, using different tau tracers at different sites, in my opinion, I think it's wrong. I don't think we should combine those studies. I don't think we have enough experience. I don't think we have the methods to be able to combine this. And we find these phantom studies to be very challenging. So, individual studies with biological variability are probably even worse. Smoothing doesn't just do anything. And deep learning methods remains to be investigated. I think I really, really wanted to get the bottom of it, to understand what is actually going on, to be able to develop methods, or even can we actually do that? We've done a lot of harmonization for the MRI studies. These are some of our papers. There are some that have just been published, which I do not have here. We are still to try to take some of those MRI methods and apply to the PET. But as I said, it was just like so much to understand in terms of the actual process in order to be able to develop these methods. There are a lot of people that are my collaborators. So I think I would like to give a lot of credit to Vivian Zhang from the Biostats at the University of Pittsburgh. She's a master's student. She's done a lot of the processing for the blood biomarker data and all the analysis. Charles and Chen, both of them PhD graduate students, and they've done a lot of processing for the PET data. A lot of my collaborators at University of Pittsburgh, PET Center, Biostats department, and all over the United States. So thank you so much.
Thank you, Dana. So sorry, I think there was a question related to your first part of the biomarker normalization. Sorry, I missed the timing to ask the question. What is the difference between quantile regression and then quantile normalization you used?
So I think the quantile regression is the quantile normalization. I think the data was ranked, and the quantile regression is that just a regular quantile regression that you would do for the skewed data.
Okay. And then there's another question from Simon. Why would smoothing be stopped by to harmonize data? My intuition is that smoothing would have no effect on harmonizing data, so I can understand the logic.
I am 100% in agreement with you, Simon. I think because of the resolution, a lot of the PET field is harmonizing to an 8 millimeter is sort of the standard way of people believe that smoothing the data to an 8 millimeter kernel would actually get rid of the differences in whatever it is in the scanner harmonization. That's the idea behind. And we actually have tried to show that that doesn't work, because in one of our PET meetings, we had a guest and somebody said, you guys should use smoothing because that should harmonize your data. And there was a PET researcher that continuously said it doesn't work. And we kind of had to prove that it doesn't actually work using phantom studies.
Simon, does that answer your question?
Yeah, thank you. Also, have you tried using combat voxel eyes in the PET data too?
No, we have not. We used combat in the I think in the superpixel combat harmonization for the MRIs, and it did pretty good. We did not use the voxel-wise harmonization in the PET. And one reason is that we work a lot with the summary measures. So we don't work that much at the voxel level per se, but we have not used it. So one thing with the combat is that we still see a lot of variability even after we apply it in the for the summary measures, especially for the PET. And I haven't quite understood why that is.
Thanks. Yeah, I'm beginning to. So some of my collaborators are working. I'm about to analyze a PET data set that's collected at multiple sites. You might even be involved in that project. So I might with Warren Taylor. I think.
Oh, okay.
So I'm gonna have questions for you later.
Yes, I am happy to answer or talk about PET. I really think that we can make contributions to this field, and I think that we as statisticians are kind of shy when it comes to interacting with the investigator and asking questions specifically about the way the data was collected, right? And how was processed and how was everything done, because it's a lot of variability. And I think just even like existing methods we can actually use and try to fix it. But one thing that I think we learned from the head study is that we should never combine these tracers, especially across sites. That is my personal opinion, and I think there are going to be people that are going to argue and disagree with me. But from what we have seen, I don't think these tau tracers specifically should be combined. So there is another question here, right? What are your thoughts on the scanner site variability impacting interventional clinical trial results? Oh, that is the whole point. That is going to be major, because specifically when you see such differences and with the tau tracers, think about the meninges, think about the off-target binding that I've shown you. That happens in one site, it doesn't happen at another site. And even if you use the same tracer, how is that going to impact? How is that going to inflate your results? Or maybe you're going to remove that for one site. Maybe it's going to get rid of the voxels that are actually important. So I think it's very, very important to have these very controlled experiments, and we do a very good job in randomized clinical trials with that. But again, in my opinion, tracer combination and trying to harmonize these tracers, I am not sure if we want to do that, because the biology is so different and it's related so much to the individual biological variability. I don't think we can separate that. We've been trying to think about ways of how to do that, but I am not sure. And these phantom studies can only capture scanner differences. So yeah, the interventional clinical trials, that is going to be a major problem when we test using these tracers.
Thank you, Dana. Also, I was thinking of the bepranemab study where there was a positive result on the tau PET and nothing on the clinical endpoint, and was wondering, seeing this, if there's some, if all the scanner noise and stuff is going to impact some of that.
I think so. I think it's going to, and as I said, there are a lot of differences even between males and females, and I think the PET, they have a lot more massaging that they are doing behind the scenes that we do in MRIs and fMRI, especially the back filtering projection and reconstruction, which is a major thing in PET, and they use different type of reconstruction methods, and I'm not sure if the reconstruction is happening at each site separately. And it also depends, because my understanding is, and again, I am not a PET specialist, my understanding is that each PET scanner comes with its own software in terms of reconstruction, and if you have different scanners, I'm not sure how much variability there is between those.
I think there's another question from Joe. So do you want to unmute yourself and then ask? Joe Lund?
Sure. The question is that I saw in one of your slides, there is a strong side effect. This is something confused me, because I think if for different hospital, different sites, if they are using the same brand of scanner, same protocol, supposedly there should be no side effect. And any reasons and any explanations for the possible underlying mechanism?
So first of all, if we talk about PET or if we talk about MRIs, so one thing I can tell you, we have a paper published, misspelled paper, where we looked at the same subjects, scan on 4 different scanners. 2 scanners were Siemens and 2 scanners were Prisma. They were both the same scanner, same manufacturer, but different, like different type in the sense. And we found differences in that. We found differences in the PET scanners. I think, and as I've seen, and as you've seen in some of my slides, even the floor that the scanner they put on, that could influence. So I know, for example, at the University of Wisconsin, when the scanner was moved from the Weizmann Center from one floor to another floor, there was a difference. In our PET center here, when we move the scanner to another room, there is also a difference, because I think every time you move the scanner has to be calibrated to that. And even if it's the same manufacturer and the same parameters, you still see differences. And the only way to see it is through those phantom studies. And even with the phantoms, we have seen differences now. The scanners that we used at Pittsburgh with the Phantom were 2 different scanners, 2 different manufacturers. So that's why we propose to take the Phantom and scan it at different sites, different centers. And that would be the only way to know the actual truth, right? If you have a Phantom. Because with the subject, even, a little bit of movement, even anything can actually influence. And think about the voxels, right? What are they, 1 by 1 by 1 in an MRI, and even like 0.5, how much, you just need a little bit of an error to do that. And processing, we process the same data, 2 different sites, exactly the same pipeline, we got different results. So I think it's, you know, as long, we don't know what the truth is. That is our main problem. We don't know what the truth is. But if we know the truth, maybe would be like, okay, we have to apply some sort of a correction. I don't know. We see that there is only 10% difference because we took this phantom and we scanned it at 5 different sites. And I think there are studies, I think Taki did studies where they took a subject to different sites, and they have shown differences, you know, around. So
Thanks for your answer.
I think we already passed 1. So if there's no more questions, so then maybe just give you a final note before you leave. So we have our next seminar schedule of 1 to 2 on August 11 by Taki Shinohara. And I think Dana mentioned multi-time about Taki. And then Taki will talk about harmonization again, but we'll focus on MRI harmonization. All right, so thank you so much, Dana, and then thank you all for joining us today, and hope to see you next time.
Thank you so much for being here, guys. Appreciate it. Thank you.
Thank you. Bye,
Thanks,
everyone.
very
Bye.
interesting.